Hello, everybody. Today I'll be presenting on beta amyloid-induced immune response found in Alzheimer's disease. So, what is Alzheimer's disease? It is characterized as a neurodegenerative disorder where it is the progression of cognitive decline. It is one of the most common forms of dementia in older people, and it is widely believed to be driven by spontaneous clustering of amyloid beta peptide into oligomers and fibrils that accumulate into plaques. So, what is beta amyloid or amyloid beta? It is a protein that is found at high levels, which is highly associated with the cognitive impairment in Alzheimer's disease. There are many different forms of beta amyloid, peptide, protein, oligomer, fibril forms. Um, it is an early indicator for Alzheimer's also, and the formation and the accumulation of beta amyloid plaques in the brain is what is characteristic for Alzheimer's disease. So the immune response to beta amyloid is primarily through the interaction between beta amyloid with microglia and Alzheimer's, and the microglia is a glial cell that is essential for immune defense in the central nervous system. And in Alzheimer's, there's a strong association between microglia-induced neuroinflammation. The first study that I looked at was looking at the immune hyperactivity of the amyloid beta plaque associated microglia in Alzheimer's disease, which basically is focusing on how beta reacts with the interaction with beta amyloid and the resulting immune response from that. And they used transgenic mouse models in the experiments in human tissue samples, and they performed multiple experiments. We'll cover two of them. And the methods that they used included LPS injection, microglia isolation, immunohistochemistry, and immunofluorescence. And for the first experiment that we'll cover, it involved LPS or PBS injection of the mice. The mice were then anesthetized and then euthanized. And then the brains were fixed in the paraformaldehyde for one day, then sucrose, and then eventually frozen in cryostat. And then we can see this resulting figure and the different mice groups that were involved were the wild type, the APP23, and the 5XFAD group, which are both transgenic mouse models to study Alzheimer's disease. And specifically, they have an accumulated presence of amyloid beta at some capacity. So these mice were challenged with LPS or PBS, and LPS is meant to induce an immune reaction, and PBS is acting as a control to ensure that the effects are from LPS and not other factors. And the first thing that I wanted to ask was, based on what we learned in class, what is IL-1 beta, if you recall, that form? And if you do remember, you would remember that it is a form of a cytokine. And then another important marker that we are involved in this experiment is IBA1, which is a microglial marker, which is an indicator of the activation of microglia. So in the first part in A, it's just indicating the mice were either injected by PBS or LPS. And then in B, we are able to see the different parts of the brain and the hippocampus, the pons, and the cortex, and either if they were treated with LPS or not, and the control. And the same thing with C here, and it was actually stained. The actual dense core plaques are stained with red, and then immunostained for IL-1 beta after the injection. And then we're able to see down in D, E, and F the activation of IBA1, IL-1 beta, and then merge together, see where there's overlap. And knowing what we know about IBA1 and IL-1 beta, what does the overlap of these molecules mean for microglia and beta amyloid presence? If you want to take a little bit of time to think about that. So, knowing that IL-1 beta is a cytokine that does respond to amyloid beta and that IBA1 is a microglial marker, it shows that amyloid beta associated microglia show increased inflammatory reactivity from the activation of IL-1 beta. Next, we have a second figure from this same study, where we look at the sections of the hippocampus from a 40-year-old early onset Alzheimer's patient, and then we see the hippocampus of an 81-year-old late onset Alzheimer's patient that were both immunostained for IBA1 and amyloid beta. And then in the second figure, for figure C, we can see the IBA1 mean gray value being compared from early onset versus late onset. And we can see from that data that both the plaque and the non-plaque region of late onset has a significantly higher IBA1 value compared to the early onset. And I just want you to think to yourself, comparing the immunofluorescence between the early onset versus late onset, what are some differences in the activation of IBA1 and amyloid beta between these two based on part A and B of this figure? So we can see that there's a pretty noticeable difference between early onset Alzheimer's compared to late onset Alzheimer's, where the amyloid beta is less concentrated in the early Alzheimer's and therefore has less IBA1 activation, meaning there's less microglia activation in this person's brain, while with late onset Alzheimer's, there's more concentration of the amyloid beta, as you can see the more bright red marking. And then there's also more surrounding IBA1 activation. So this shows that the late onset Alzheimer's has more concentration and therefore more microglial activation in response to amyloid beta. So next, I looked at another study, and the study was basically trying to observe how the NALP3 inflammasome reacts to amyloid beta presence and its contribution to neuroinflammation in Alzheimer's disease. And the molecules that were involved in this experiment were reverse amyloid beta, which acts as a negative control in studies inducing amyloid beta uh, inflammation. And then ATP is acting as a positive control and is an activator, another known activator for NALP3. And then we have caspase 1, which is an enzyme that indicates that it is something's being activated from the inflammasome. And we can see from figure A and B that the ELISA is showing the release of IL-1 beta from the wild type primary mouse microglia, meaning it's coming directly from the mouse. And we can see that the amyloid beta levels are exceptionally high at hour 6. Well, regarding for IL-1. And then when it comes to the microglia cell line, the immortalized mouse microglia, um, we can see that the amyloid beta levels are also high, but you can see it at hour 3, 6, and after 24 hours, you can see that IL-1 beta is high. And then for part C, we can see from the microscopy and the flow cytometry that there is activated capsase in amyloid beta and ATP. And so we have a question based on figure 1C, seeing what is being activated from amyloid beta and the ATP treatments. What does the presence of capsase 1 indicate? And remember, this is an indicator of capsase 1 is an enzyme activated from inflammasome, if you want to take a little bit of time to think over that. So knowing what capsase 1 is, since it is an enzyme that indicates the activation of inflammasome, it shows that there is amyloid beta with when they're indicating that there's amyloid beta that activates the NLP3 inflammasome and therefore triggers the inflammation within the cells. Specifically, in this case, it is measured in the microglia cells. So the overall findings from these studies are only a couple out of many different studies that investigates the many immune responses from beta amyloid plaque accumulation and and these studies all further help characterize Alzheimer's disease in general. By knowing the types of immune cells involved and that are activated, it is essential to help find new treatments for the disease. And now we know that microglia has a very significant role in the inflammation of amyloid beta plaques and accumulation, and cytokines like IL-1 beta are involved, along with IBA1 activation that both indicate higher activity when amyloid beta is present. We also know that another aspect of Alzheimer's disease from the amyloid beta presence is that NALP3 inflammasome is activated from from amyloid beta and is seen to be activated by the presence by caspase 1 enzyme, especially when you recall back to the immunofluorescent imagery of the cells. And then IL-1 beta is higher in concentration in general when amyloid beta is present when microglia activates. Thank you for listening to my presentation, and I hope you have a little bit more understanding of Alzheimer's disease and the role of amyloid beta.